So here's something worth sitting with for a moment. You, right now, lying wherever you are, breathing without thinking about it, your heart beating on its own, your eyes scanning or closing. You are the product of roughly 4 billion years of unbroken biological engineering. 4 billion years. Not a single link in that chain was ever broken. Every one of your ancestors, stretching back through time in an unimaginable line, survived long enough to reproduce. Every single one. Through ice ages and asteroid impacts and famines and plagues and predators and sheer dumb luck, that line held. And here you are. The latest draft of a very, very old blueprint. That's what we're going to talk about tonight. Not all at once. Not in a rush. We're going to walk through it slowly. The way you'd walk through an old museum late at night when nobody else is around and you can just stand in front of each display for as long as you want. The story of how we got here. How a planet covered in molten rock and toxic gas eventually produced a creature that writes poetry, builds bridges, worries about the future, and sometimes lies awake at night wondering where it all came from. Let's start at the very beginning. And I mean the very beginning, before anything we'd recognize as life existed at all. About 4.5 billion years ago, Earth was not a hospitable place. Picture a world with no oxygen in the atmosphere, no oceans, at least not at first. The surface was mostly molten or barely solidified rock, constantly bombarded by debris left over from the formation of the solar system. Meteorites were slamming into the surface so regularly that the planet couldn't even cool down properly. The sky, if you could call it that, was thick with carbon dioxide, methane, ammonia, water vapor, a choking, scalding blanket of gases that would kill any modern organism in seconds. There was no ozone layer, so ultraviolet radiation from the young sun poured down unfiltered. It was, by any standard we'd recognize, hell. And yet, somewhere in that chaos, chemistry started doing something remarkable. Here's the thing about life, and this is one of those questions that still keeps scientists up at night. We don't know exactly how it started. We have very good hypotheses. We have experiments that demonstrate key steps. But the precise sequence of events that turned non-living chemistry into the first self-replicating molecule, that's still one of the great open questions in all of science. And honestly, that makes it more interesting, not less. What we do know is that by about 3.8 billion years ago, maybe even a little earlier, something was alive on Earth. We know this because of chemical signatures in ancient rocks, carbon isotope ratios that suggest biological processing. The oldest definitive fossils we've found, these layered structures called stromatolites, date to about 3.5 billion years ago. Stromatolites are built by colonies of microorganisms, mostly cyanobacteria, layering sediment and minerals over time. You can actually still find living stromatolites today, in a few places around the world. Shark Bay in Western Australia is one of the most famous spots. They look like lumpy underwater cushions, nothing dramatic. But when you realize you're looking at essentially the same kind of structure that dominated Earth for billions of years, it hits different. So life appeared surprisingly fast, cosmically speaking. Earth formed around 4.5 billion years ago. The heaviest bombardment tapered off maybe 4 billion years ago, and within a few 100 million years, which sounds like a lot, but in geological terms it's practically overnight, something was alive. That speed has led some researchers to argue that life might be almost inevitable given the right chemistry and conditions. Others think we just got extraordinarily lucky. The honest answer is we don't know yet. But let's talk about what those earliest organisms actually were, because this matters for understanding everything that comes after. The first life forms were prokaryotes, single-celled organisms without a nucleus. Think bacteria. Incredibly simple by our standards, but don't let that fool you. A single bacterial cell is still an astonishingly complex chemical system. It has a membrane that selectively lets things in and out. It has DNA that encodes instructions. It has molecular machinery that reads those instructions and builds proteins. It can harvest energy from its environment. It can reproduce. Each one of those capabilities represents an engineering problem that took, well, we're not sure how long it took, but it happened. And here's what's really clever about these early organisms. They figured out how to eat, essentially, in a world that had no oxygen. The earliest metabolic strategies were anaerobic. They didn't use oxygen because there wasn't any free oxygen to use. Some of them probably fed on simple chemical compounds dissolved in the water around hydrothermal vents, those cracks in the ocean floor where superheated mineral-rich water gushes up from below. The energy gradients around those vents, hot meeting cold, acidic meeting alkaline, may have provided the spark that got the whole thing going. A scientist named Mike Russell proposed this idea in the late 1980s, and a version of it has gained a lot of traction since then. The idea is that natural mineral structures in those vents could have acted as tiny compartments, almost like primitive cells, where chemical reactions could concentrate and build complexity. It's elegant because you don't need some single miraculous event. You need a sustained environment where chemistry can keep experimenting. Now, for an incredibly long time, and I want you to really feel how long this is, life on Earth was just microbes. Single-celled organisms floating, dividing, metabolizing, dying, evolving in tiny increments. We're talking about roughly 2 billion years. 2 billion. If you compressed all of Earth's history into a single 24-hour day, life appears around 4 in the morning, and then nothing but single-celled organisms until sometime around 2 or 3 in the afternoon. That's most of the day, most of the story. And during that enormous stretch, something was happening that would change the planet forever. Cyanobacteria, those little photosynthetic microbes that build stromatolites, figured out a trick. They learned to use sunlight to split water molecules and harvest the energy, releasing oxygen as a waste product. Oxygenic photosynthesis. And this was, without exaggeration, the most consequential metabolic innovation in the history of life on Earth. Because oxygen is a highly reactive gas. It wants to combine with things. And for the first billion years or so of cyanobacterial activity, all that oxygen they were producing got soaked up. It reacted with dissolved iron in the oceans, creating massive deposits of iron oxide that settled to the seafloor. Those deposits are called banded iron formations, and they're the source of most of the iron ore we mine today. Think about that. The steel in the car you drive, the beams in the building you work in. That iron was rusted out of ancient oceans by the metabolic waste of organisms that lived 2.5 billion years ago. The deep past is literally holding up the structures of your daily life. Eventually, though, the oxygen sinks got saturated. The dissolved iron was used up, the reactive minerals on land were oxidized, and free oxygen started accumulating in the atmosphere. This event, which unfolded roughly between 2.4 and 2 billion years ago, is called the Great Oxidation Event, and it was both a triumph and a catastrophe. A triumph because oxygen opened up entirely new metabolic possibilities. Aerobic respiration extracts far more energy from food than anaerobic methods, which eventually made complex, energy-hungry life possible. A catastrophe Because for the anaerobic organisms that had dominated Earth for over 1 billion years, oxygen was poison. It destroyed their cellular machinery. The Great Oxidation Event was, in a very real sense, the first mass extinction, and it was caused by life itself, by pollution, if you want to think of it that way. The cyanobacteria didn't mean to do it. They were just living, doing their thing, splitting water, and the waste product of their success reshaped the entire planet. There's a lesson in that, isn't there? The most transformative changes aren't always intentional. Sometimes they're side effects, so now we're in a world with oxygen in the atmosphere, but still a world of microbes. And the next great leap, and this one is genuinely wild, happened somewhere around 1.5 to 2 billion years ago, though the exact timing is debated. One prokaryote, probably an archaeon, engulfed another prokaryote, probably a bacterium capable of aerobic respiration. But instead of digesting it, the host cell kept it alive inside. The engulfed bacterium kept doing its thing, burning oxygen for energy, and the host cell benefited from that energy. Over time, the engulfed cell lost its independence. It transferred most of its genes to the host's genome. It became an organelle. It became the mitochondrion. You have mitochondria in almost every cell of your body right now. Billions of them. They still have their own DNA, their own membranes, their own ribosomes, remnants of their former life as free-living bacteria. Every time you take a breath, you're feeding oxygen to what are essentially ancient bacterial passengers that moved in 2 billion years ago and never left. This theory, called endosymbiosis, was championed most famously by Lynn Margulis, who first proposed it rigorously in 1967. And she had a rough time of it. Her paper was rejected by about 15 journals before it was finally published. The scientific establishment thought the idea was too radical, too strange. Cells don't just swallow other cells and form partnerships. That's not how evolution works, they said. But Margulis was right. The evidence is now overwhelming, and a similar event happened later, with cyanobacteria being engulfed to become chloroplasts, the organelles that carry out photosynthesis in plant cells. So plants are running on captured cyanobacteria, and you and I are running on captured aerobic bacteria. We're all chimeras, assemblages of once independent organisms that learn to cooperate. And this is where it gets interesting, because that merger, that endosymbiotic event, is what made eukaryotic cells possible. Eukaryotes are cells with a nucleus, internal membranes, organelles, all that complexity. You're made of eukaryotic cells. So is every animal, every plant, every fungus. And the energy boost provided by mitochondria is what allowed eukaryotic cells to get bigger, to maintain larger genomes, to support more complex internal structures. Without that ancient partnership, complex life as we know it probably never happens. So now we have eukaryotic cells, and we're somewhere around 1.5 billion years ago, give or take. And for another long stretch, hundreds of millions of years, life is still mostly single-celled. Eukaryotic microbes are diversifying, exploring new ecological niches, getting more sophisticated in their own right. Some of them are developing sexual reproduction, which is a whole story in itself. The ability to shuffle and recombine genetic material between individuals, which dramatically accelerates the pace of evolution by creating new combinations of traits for natural selection to work on. But the next really big transition, the one that starts to feel like it's heading toward us, is multicellularity. The moment when individual cells started sticking together, cooperating, specializing, forming something larger than themselves. And here's what's fascinating. Multicellularity didn't evolve just once. It evolved independently dozens of times across different lineages: animals, plants, fungi, various algae. They all arrived at multicellularity separately, which tells you something important. It must be a really good strategy. When the conditions are right, evolution keeps finding its way to the same solution. The earliest evidence of multicellular life is still being pushed back as new fossils are discovered, but we have reasonably confident examples from around 1.2 billion years ago. Red algae fossils from Arctic Canada that show differentiated cells, meaning cells that had taken on specialized roles within the organism. That's the key insight about multicellularity. It's not just about cells clumping together. It's about cells giving up their individual autonomy, specializing, some becoming structural, some becoming reproductive, some handling nutrient transport, and the whole assembly becoming more than the sum of its parts. It's cooperation at the cellular level, and it requires trust, in a sense. Each cell carries the full genome, the full set of instructions, but it only reads the parts relevant to its job. A muscle cell has the same DNA as a nerve cell, but they behave completely differently because different genes are switched on and off. And that brings us to a stretch of time that paleontologists sometimes call the boring billion, roughly the period from about 1.8 billion to around 800 million years ago. It's called boring because, compared to what came before and what came after, not a lot seems to change in the fossil record. Oxygen levels were relatively stable but still much lower than today. Life was mostly microbial, with some early multicellular experiments. The continents were drifting around, assembling and breaking apart. It was a long, quiet middle chapter. But quiet doesn't mean nothing was happening. Beneath the surface, in the genomes of those early eukaryotes, the genetic toolkit for complex animal life was being assembled, piece by piece. The genes for cell adhesion, for cell signaling, for programmed cell death, all the molecular machinery you need to build a body. Much of it was evolving during this supposedly boring interval. It's like the long years an engineer spends in a workshop, tinkering, prototyping, testing small components before the big machine is finally assembled and switched on. And that machine was about to switch on. Because around 800 million years ago, the world started to change dramatically. Oxygen levels began to rise again. The supercontinent Rodinia was breaking apart, opening new ocean basins, altering ocean chemistry. And then, something happened that sounds almost too dramatic to be real. The entire planet, or very nearly the entire planet, froze over. Snowball Earth. Ice sheets extending from the poles all the way to the equator. Oceans freezing over. Global temperatures plummeting. It happened more than once, these extreme glaciations, roughly between 720 and 635 million years ago. And when the ice finally melted, when volcanic carbon dioxide built up enough in the atmosphere to trigger a massive greenhouse warming, the world that emerged was chemically and ecologically transformed. Oxygen surged. Nutrients flooded into the oceans from newly exposed weathered rock. And in that strange, thawed, oxygen-rich aftermath, something extraordinary began to stir in the shallow seas. Something with bodies. Something with structure. Something you could actually see with your naked eye, if you'd been there, wading in those shallow, sun-warmed waters around 570 million years ago. And what you would have seen would have stopped you in your tracks, because nothing like it had ever existed before on this planet. These were the Ediacaran organisms, named after the Ediacara Hills in South Australia, where their fossils were first recognized for what they really were. And they are genuinely strange. Imagine soft, quilted forms lying on the seafloor or anchored upright in the sediment like fleshy fronds, Some of them looked like ribbed discs. Some looked like feathery leaves. Some looked like nothing you've ever seen in any biology textbook, because they don't neatly fit into any modern category of life. There's one called Dickinsonia, a flat, oval thing with segmented ridges radiating out from a central line, almost like a bath mat. It could be a few centimeters across or over a meter wide. For decades, scientists argued about what it even was. A giant amoeba? A lichen? A jellyfish? A failed experiment in multicellular life that left no descendants? It wasn't until 2018 that a team led by Ilya Bobrovsky, working in Russia, found preserved cholesterol molecules, cholesteroids, in Dickinsonia fossils from the White Sea region. Cholesterol is an animal biomarker. That settled it, or mostly settled it. Dickinsonia was an animal, one of the earliest animals we know of. But here's the thing about the Ediacaran world. It was an animal world unlike anything that came after. These creatures had no shells, no skeletons, no teeth, no claws. As far as we can tell, most of them had no mouths. They seem to have absorbed nutrients directly through their skin, or maybe through symbiotic relationships with microbes. There were no predators. Nothing was chasing anything. Nothing was biting anything. It was, in a way, a peaceful world. Some paleontologists have called it the Garden of Ediacara, and there's something almost wistful about that phrase, because it didn't last. The Ediacaran period runs from about 635 to 538 million years ago, and during that span, you see these soft-bodied communities flourishing on the seafloor. They lived in a world where the ocean chemistry was still very different from today. Oxygen was rising, but unevenly distributed, and there were probably vast zones of anoxic water lurking in the deep ocean. The organisms that thrived were the ones adapted to that particular chemical landscape, and they were doing something genuinely new. They were building bodies with differentiated tissues. They were experimenting with bilateral symmetry, having a left side and a right side. Some of them, like a tiny worm-like creature called Kimberella, seem to have been actively grazing on microbial mats, leaving scratch marks on the seafloor that you can still see preserved in the rock. Kimberella had a front end and a back end, a top and a bottom. It moved with purpose. That's a big deal. That's the beginning of something. But most of the Ediacaran organisms disappeared. They vanished from the fossil record right around the boundary between the Ediacaran and the Cambrian, roughly 538 million years ago. What happened to them is one of the great open questions. Some may have been outcompeted by the new, more active animals that were about to appear. Some may have been eaten by them. Some may have simply been unable to cope with changing ocean conditions. The Garden of Ediacara was plowed under, and something wilder, fiercer, and far more dynamic took its place. And this is where the story gets fast, bewilderingly fast by geological standards. It's called the Cambrian explosion, and even though scientists have been studying it for over a century, it still has the power to astonish. In a span of maybe 20 to 25 million years, which sounds like a lot, but remember, life had been almost entirely microbial for over 3 billion years before this. Nearly all the major animal body plans that exist today appeared in the fossil record. Arthropods, mollusks, chordates, the group that includes us. Echinoderms, like starfish, annelid worms. Cnidarians, suddenly, or what passes for suddenly in deep time, the oceans were full of animals with hard shells, jointed legs, compound eyes, guts, gills, nervous systems, and, crucially, the ability to chase, capture, and eat each other. Think about what that means. For billions of years, life had been mostly cooperative, or at least passively competitive. microbes jostling for nutrients, algae competing for light. But now, for what seems like the first time, you had active predation as a major ecological force. And predation changes everything. It creates an arms race. If something is trying to eat you, you need armor, or speed, or camouflage, or the ability to burrow, or eyes to see it coming. And if your prey develops armor, you need stronger jaws, or claws, or a new hunting strategy. This feedback loop, sometimes called the evolutionary arms race, is one of the most powerful engines of diversification in the history of life. It drives complexity. It drives innovation. It drives the relentless, restless creativity of natural selection when the stakes are life and death, every single day. The best window we have into this Cambrian world is a place called the Burgess Shale, high up in the Canadian Rockies of British Columbia. It was discovered in 1909 by Charles Doolittle Walcott, who was the secretary of the Smithsonian Institution, basically the most powerful scientist in America at the time. Walcott was riding along a mountain trail near the town of Field when, according to the famous story, his horse stumbled on a slab of shale, and he looked down and saw fossils of extraordinary preservation. Soft tissues, appendages, guts, even the contents of the last meal, all pressed into fine-grained mudstone that had been deposited on the floor of a Cambrian sea about 508 million years ago. Now, the story of Walcott's horse stumbling is probably embellished. Walcott's own field notes suggest a more systematic search. But the discovery was real, and it was staggering. Over the following years, Walcott excavated tens of thousands of specimens from the quarry. And here's where the human drama comes in, because Walcott, despite being a brilliant collector, was in many ways a product of his time. He tried to fit every one of those bizarre Cambrian creatures into existing categories. He shoehorned them into known phyla, into familiar groups. He didn't fully appreciate just how strange they were, how many of them represented body plans that no longer exist anywhere on Earth. It took decades for other scientists to revisit Walcott's collection and realize what he'd missed. In the 1960s and 70s, a British paleontologist named Harry Whittington, along with his graduate students Simon Conway Morris and Derek Briggs, began a painstaking re-examination of the Burgess Shale fossils at the Smithsonian. And what they found shook paleontology. Creatures like Anomalocaris, a meter-long predator with grasping appendages and a circular mouth like a pineapple ring, unlike anything alive today. Hallucigenia, which was so bizarre that the first reconstruction had it upside down and backwards. What they thought were legs turned out to be spines on its back, and what they thought were tentacles on its back turned out to be its legs. Opabinia, with 5 eyes and a forward-facing nozzle-like proboscis. These animals were real. They lived. They swam and crawled and hunted in seas that covered what is now a mountain range in Canada. Stephen Jay Gould wrote a famous book about this in 1989 called Wonderful Life, arguing that the Cambrian explosion represented a period of maximum evolutionary experimentation, and that many of those experiments were snuffed out not because they were inferior, but because of sheer contingency, bad luck, wrong place at the wrong time. His argument was that if you could rewind the tape of life and play it again, you'd get a completely different set of survivors, and maybe nothing like humans would ever appear. Conway Morris, who had done much of the original descriptive work, later pushed back hard against this, arguing that convergent evolution, the tendency for natural selection to find the same solutions over and over, meant that something like intelligence, something like eyes, something like active predation would probably evolve no matter how many times you replayed the tape. That debate is still alive, still unresolved, and honestly, it's one of the deepest questions you can ask about our own existence. Were we inevitable, or were we a fluke? But let's stay in the Cambrian for a moment longer, because there's one particular group of animals from this period that matters to you more than any other, and you might not guess which one. They're small. They're soft-bodied. They're easy to overlook. They're called chordates, animals with a flexible rod running along their back, called a notochord. The earliest chordates in the Cambrian were tiny, wriggling, fish-like creatures just a few centimeters long. One of the most famous is Pikaia from the Burgess Shale, though there's now debate about whether Pikaia is a true chordate or something slightly more primitive. A better candidate might be Hyquicthus from the Chengjiang fossil beds in Yunnan province, China. Another spectacular Cambrian Lagerstätte, actually a bit older than the Burgess Shale, about 518 million years ago. Hyquicthus was barely an inch long. It had a notochord, a dorsal fin, and what appear to be rudimentary vertebrae, tiny cartilaginous structures along its back. It may be one of the earliest vertebrates, one of the earliest members of the lineage that would eventually produce fish, amphibians, reptiles, mammals, and you lying here right now, listening to this. That's the thing that gets me about the Cambrian. Everything was small and strange and new, and the ancestors of every major animal group alive today were right there, swimming alongside creatures that would leave no descendants at all. The winners and the losers, side by side, and no way to tell them apart at the time. No way to know that the little wriggling thing with the stiff rod in its back was going to matter more, in the long run, than the magnificent Anomalocaris with its crushing appendages and its compound eyes. And those eyes, let's talk about those for a second, because the evolution of eyes is one of the most remarkable stories in all of biology. In the Cambrian, we see the first really sophisticated visual systems appearing. Trilobites, those iconic Cambrian arthropods with their segmented bodies and their crescent-shaped heads, had compound eyes made of calcite crystal, actual mineral lenses. Some trilobite species had thousands of individual lenses packed into each eye, giving them a wide field of vision across the seafloor, and the optics were extraordinary. In some species, each lens was shaped as a doublet, correcting for spherical aberration in a way that would make an optical engineer nod with appreciation. These animals solved a physics problem with biological engineering 500 million years ago. The British-Australian scientist Andrew Parker proposed what he called the light switch hypothesis. The idea that the evolution of sophisticated eyes was itself a trigger for the Cambrian explosion. Once predators could see, the entire ecological game changed. Prey needed to hide, to camouflage, to develop hard shells that couldn't be spotted easily or that resisted attack. The visual arms race cascaded through every ecosystem, driving rapid diversification. It's a compelling idea, though probably not the whole story. Rising oxygen, changing ocean chemistry, the evolution of key developmental genes. All of these played a role. The Cambrian explosion was probably not one event with one cause, but a cascade of reinforcing feedbacks, each amplifying the others. And after the Cambrian came the Ordovician, and this is when life really began to fill in the ecological spaces that the Cambrian had opened up. If the Cambrian was the grand opening, the Ordovician was the build-out. More species, more niches, more complexity. Marine biodiversity tripled. Reef ecosystems expanded, and our own lineage, the vertebrates, began to diversify in earnest. The first jawless fish, creatures like Arundaspis and Sacabambaspis, appeared, armored with bony plates, filter feeding in shallow seas. They didn't have jaws yet. That innovation was still coming. But they had something important: bone, mineralized tissue, a skeleton that could fossilize, that could support larger body sizes, that could store minerals like calcium and phosphorus right there inside the body, available when needed. Think about that for a second. Before bone, animals were soft, or they had exoskeletons made of chitin or calcium carbonate, external armor. Bone was different. It was internal scaffolding, living tissue threaded with blood vessels, capable of growing with the animal, remodeling itself under stress, healing when it broke. And it gave vertebrates something that would prove to be an extraordinary long-term advantage: an internal frame that could be modified, elongated, branched, and reshaped by evolution into an almost infinite variety of forms. Every skeleton you've ever seen, the delicate bones of a sparrow's wing, the massive femur of an elephant, the tiny ossicles in your own middle ear, all of it traces back to this Ordovician innovation. Bone was the platform, but those early jawless fish, the ostracoderms as they're sometimes called, were humble creatures. Picture something the size of your hand, maybe a bit larger, shaped vaguely like a flattened tadpole, covered in bony plates across its head and the front of its body, but with a more flexible tail for swimming. No jaws, just a round or slit-like mouth that could suck in water and filter out tiny food particles. They hugged the bottom of shallow seas, probably sifting through sediment. Not exactly apex predators, but they were us, in a sense. They were on our branch of the tree, and they were quietly diversifying, experimenting with different body shapes and different arrangements of those bony plates, while the invertebrate world around them, the trilobites, the brachiopods, the nautiloid cephalopods with their long, straight shells, dominated the Ordovician seas. Now, the Ordovician is one of those periods that doesn't get the fame it deserves. It lasted from about 485 million years ago to around 443 million years ago, and during that span, marine life underwent what paleontologists call the Great Ordovician Biodiversification Event. It's a mouthful, but the idea is straightforward. The number of marine families roughly tripled. New reef ecosystems appeared, built not just by sponges and microbial mats, but by corals and bryozoans. Crinoids, those beautiful feathery animals sometimes called sea lilies, carpeted the seafloor. Graptolites drifted in the open water. And the cephalopods, those relatives of modern squid and octopuses, became the top predators of the Ordovician Ocean. Some of them, the orthocone nautiloids, had straight conical shells that could reach several meters in length. Imagine encountering one of those, a jet-propelled predator the length of a car, cruising through clear Ordovician waters. For a small, jawless fish minding its own business on the seafloor, that was a very real threat. And then, the Ordovician ended. Badly. Around 443 million years ago, Earth experienced one of the 5 great mass extinctions in its history. The late Ordovician extinction wiped out perhaps 85% of all marine species. The cause, as best we can tell, was a severe ice age. The supercontinent Gondwana had drifted over the South Pole, and massive glaciers formed, locking up water and dropping sea levels dramatically. Shallow seas, the very habitats where most of that extraordinary Ordovician diversity lived, simply drained away. The continental shelves that had been teeming with life were exposed as dry land. And as the glaciers grew, ocean circulation patterns shifted, pulling cold, Oxygen pour water up from the deep, and suffocating organisms adapted to warmer, oxygen-rich conditions. It came in two pulses, this extinction, separated by perhaps 1,000,000 years. The first pulse hit as glaciation began and sea levels dropped. The second hit as the glaciers melted and sea levels rose again. But now the ocean chemistry had changed. Anoxic water spread across the shelves, and species that had survived the first pulse succumbed to the second. It's worth pausing here to think about what mass extinctions actually mean. We'll encounter several more as we move through this story, and each one reshapes the trajectory of life on Earth in profound ways. An extinction isn't just subtraction. It's not just fewer species. It's the removal of ecological incumbents, the dominant groups that had been holding down the fort, occupying the best niches, suppressing competitors. When those incumbents disappear, the survivors find themselves in a world with empty space. And evolution, given empty space, gets creative fast. The recovery after a mass extinction is often where the most dramatic innovations appear, because suddenly there's room to experiment. After the Ordovician extinction, life recovered through the Silurian period, and this is when something happened that would change our lineage forever. Somewhere in the Silurian seas, around 430 million years ago, give or take, a group of fish evolved jaws. And here's what's really clever about how it happened. Jaws didn't appear from scratch. They were repurposed. The leading hypothesis, which goes back to the work of anatomists in the 19th century and has been refined with modern embryological and genetic evidence, is that jaws evolved from gill arches, those bony or cartilaginous supports that held open the gill slits in jawless fish. Evolution took one of the front pairs and gradually modified them, hinging them, adding muscle, turning a breathing structure into a grasping structure. It's a beautiful example of what biologists call exaptation, where a feature that evolved for one function gets co-opted for a completely different one. And jaws changed everything. A fish with jaws could bite. It could grab prey, crush shells, tear flesh. It could exploit food sources that were completely inaccessible to a filter feeder. The jawed fish, the gnathostoms, radiated rapidly. And you are a gnatostom. Every mammal, every bird, every reptile, every amphibian, every bony fish, every shark, all gnathostoms. That Silurian jaw innovation is one of the most consequential in vertebrate history. You use yours every time you eat, every time you speak, every time you yawn. The Silurian also saw another momentous development, though it was happening not in the ocean, but on land. Plants had been creeping onto shore since at least the Ordovician. Simple things, probably resembling liverworts, hugging wet ground near the water's edge. But during the Silurian, around 420 million years ago, the first vascular plants appeared. Plants with internal plumbing, tubes that could carry water upward from roots and sugars downward from leaves. This was the engineering breakthrough that allowed plants to grow tall, to stand upright, to colonize drier ground farther from the water. A plant without vascular tissue is limited to a few centimeters in height, dependent on direct contact with moisture. A plant with xylem and phloem can be a tree. Not yet. Not in the Silurian. The first vascular plants were still small, just a few centimeters tall. Things like Cooksonia with its simple forking stems and tiny spore-bearing capsules at the tips. But the architecture was in place. The engineering problem of how to be a tall organism on dry land, how to resist gravity without the buoyancy of water, how to transport fluids internally, how to reproduce without swimming sperm, was being solved, piece by piece. And here's where I want you to appreciate something about the colonization of land. because it's easy to take for granted. We live on land. It feels normal. But for the first 3 billion years of life on Earth, land was essentially a dead zone. Bare rock, sand, and microbial crusts. No soil. Soil requires organic matter, requires roots to break rock, requires fungi and bacteria to decompose dead tissue. The land surface of the early Earth was more like Mars than anything you'd recognize. Moving onto land meant solving a completely different set of engineering problems than life had ever faced. In water, you're supported. On land, you need structural rigidity. In water, gas exchange happens across your whole surface. On land, you need specialized structures, lungs, stomata, and you need to prevent yourself from drying out. In water, reproduction can involve releasing eggs and sperm into the current. On land, you need new strategies. Every single one of these problems had to be solved, and they were solved independently by different lineages: plants, fungi, arthropods, and eventually vertebrates, each figured out their own versions. Arthropods were actually among the first animals to follow plants onto land. By the late Silurian and into the Devonian, millipedes, scorpion-like arachnids, and early insects were living on shore. Some of the earliest evidence comes from fossilized trackways and body fossils found in Scotland and other localities. These pioneers were small, but they were establishing the base of terrestrial food webs, eating decaying plant matter, eating each other, beginning the cycles of nutrient turnover that would eventually build real soil. But the Devonian period, from about 419 to 359 million years ago, this is the one paleontologists sometimes call the Age of Fishes, and for good reason. The oceans were dominated by an extraordinary diversity of fish. Sharks and their relatives appeared and diversified. Bony fish split into two great lineages: the ray-finned fish, which would eventually become the vast majority of fish species alive today, and the lobe-finned fish, which had muscular, fleshy fins supported by bones. And it's the lobe-finned fish that matter most to our story, because one group of lobe-finned fish gave rise to all land vertebrates, to you. The Devonian is where we find some of the most famous transitional fossils in all of paleontology. Tiktaalik, discovered in 2004 by Neil Shubin, Ted Deichler, and Farish Jenkins on Ellesmere Island in the Canadian Arctic, is perhaps the most celebrated. Shubin has written beautifully about the discovery, how they deliberately targeted Devonian-aged rocks in the Arctic because geological maps told them these were once tropical river deltas, exactly the kind of shallow, vegetation-choked waterways where a fish might benefit from being able to prop itself up, to push its head above the surface, to navigate through tangled plant matter. And that's exactly what Tiktaalik could do. It had a flat, crocodile-like head. It had a neck. Fish don't have necks, but Tiktaalik did, meaning it could move its head independently of its body. Its front fins contained bones that correspond to the humerus, radius, and ulna in your own arm. It had a wrist. It could do something like a push-up. It was a fish, undeniably a fish, but it was a fish that was solving the problem of living in shallow, swampy water by evolving features that would later prove essential for walking on land. And Tiktaalik wasn't alone. It sits in a sequence of fossils that beautifully illustrate the transition from water to land: Eusthenopteron, more fully aquatic; Panderichthys, a bit more flattened, a bit more adapted to shallow water; Tiktaalik in the middle; and then Acanthostega and Ichthyostega, which had actual limbs with digits, actual fingers and toes, but still lived mostly in water. Acanthostega is particularly fascinating because when Jenny Clack, a remarkable paleontologist at Cambridge, who essentially revived the study of this transition, re-examined its fossils in the late 1980s and 90s. She found that it had 8 digits on each limb, that its limbs weren't really suited for walking on land, and that it still had internal gills. It was a tetrapod, a 4-limbed vertebrate that was still fundamentally aquatic. The limbs evolved in water, probably for navigating dense vegetation in shallow swamps, for bracing against currents, maybe for ambush predation. Walking on land came later. The tools evolved for one purpose and were then co-opted for another, just like jaws from gill arches. Jenny Clack's work deserves a moment here, because her story is one of those cases where a single person's persistence transforms an entire field. When she began studying early tetrapods in the 1980s, the transition from fish to land vertebrates was considered a somewhat stale topic. The conventional wisdom, based on limited fossils, was that it was fairly well understood. Clack disagreed. She traveled to Greenland to recollect fossils of Acanthostega from sites that had been visited decades earlier, and what she found overturned the old narrative completely. The idea that tetrapods evolved legs in order to walk on land, that tidy story, fell apart. Legs came first, in water. The land came after. Clack continued working on this transition for decades, mentoring students, publishing prolifically, until her death in 2021. The field she left behind was unrecognizable from the one she'd entered, and that's largely because she had the stubbornness to look at old fossils with new eyes and the courage to say the textbooks were wrong. So by the late Devonian, somewhere around 370 million years ago, the first tetrapods were hauling themselves through swampy margins, breathing air with rudimentary lungs that, like so many innovations, had actually evolved earlier in fish as swim bladders or supplementary breathing organs for oxygen-poor water. And the world they were crawling into was itself transforming, because the Devonian was also when forests first appeared on Earth. Early trees like Archaeopteris, which could grow to 30 meters tall, were spreading across the continents, their roots breaking rock into soil, their leaves pulling carbon dioxide from the atmosphere, their fallen trunks damming streams and creating exactly the kind of swampy, complex freshwater habitats where a lobe-finned fish might benefit from having sturdy fins. There's a lovely feedback loop here. The forests were creating the environments that selected for the very adaptations that would allow vertebrates to eventually leave the water entirely. The land was building itself a welcome mat. But the Devonian, like the Ordovician before it, ended with catastrophe. Actually, with a series of catastrophes. The late Devonian extinction was not a single sharp event, but a prolonged crisis stretching over perhaps 20 million years, with several severe pulses. The Kellwasser events, around 372 million years ago, and the Hagenberg event, around 359 million years ago, devastated marine ecosystems. Reef communities collapsed and would not fully recover for over 100 million years. The great armored placoderm fish, the dominant predators of the Devonian seas, creatures like Dunkleosteus, which could be 6 meters long with a bite force rivaling a large alligator, vanished entirely. The causes are still debated. Massive volcanic eruptions, changes in ocean chemistry, the spread of land plants altering weathering rates and nutrient runoff into the oceans, probably some combination. But the result was clear. The world that emerged into the Carboniferous period was a different place, with different rulers, and our tetrapod ancestors, those swamp-dwelling pioneers with their stubby limbs and gulping lungs, were among the survivors who would inherit it. And what they did with that inheritance in the vast, steaming forests of the Carboniferous is one of the most extraordinary chapters in the whole story of life. Because the Carboniferous, which stretches from about 359 to roughly 299 million years ago, was a world that would have looked almost alien to us, and yet was building, piece by piece, the biological toolkit that you carry around inside your body right now. Let's start with the forests, because the forests are everything in this period. If you could somehow stand at the edge of a Carboniferous swamp, and you'd want to be careful where you stepped, you'd be surrounded by trees, but not trees like anything you've ever seen. These weren't oaks or pines or maples. The dominant trees of the Carboniferous were lycopsids, giant club mosses with names like lepidodendron and sigillaria, and they were bizarre. Lepidodendron could reach 40 meters tall, which is taller than a 10-story building. But here's the strange part. Its trunk was mostly bark. The interior was surprisingly hollow, almost like a tube. It grew fast, branched only at the very top in a sort of crown, reproduced with spores, and then died. Its entire life cycle might have played out in just 10 to 15 years. These weren't the slow, patient hardwoods you think of when you picture an ancient forest. They were more like weeds that happened to be enormous, and they grew in such profusion, in such dense, waterlogged stands, that when they fell, the swamp water preserved their trunks before they could fully decompose. And this is where one of the most consequential details in Earth's history comes in. Think about what happens when a tree dies today. Fungi and bacteria break it down. They consume the cellulose, the lignin, the complex carbon compounds, and they release that carbon back into the atmosphere as carbon dioxide. It's a cycle. But in the Carboniferous, there's strong evidence that the microorganisms capable of efficiently breaking down lignin, the tough structural polymer that gives wood its rigidity, hadn't fully evolved yet. Or at least, they weren't nearly as effective as modern wood-rotting fungi. Lignin had been an evolutionary innovation of the Devonian, a molecule that let plants grow tall by making their cell walls rigid, and for tens of millions of years, nothing in the microbial world had caught up. Nothing could eat it properly. So what happened? The trees grew, they fell, they piled up, and they didn't rot. Layer after layer after layer of dead plant material accumulated in those swamps, compressed under sediment, cooked slowly by geological heat and pressure over millions of years, and turned into coal. This is where coal comes from. The coal deposits that powered the Industrial Revolution, that drove the steam engines and the factories and the railroads that reshaped human civilization. Those deposits are literally the compressed, fossilized remains of Carboniferous swamp forests. The word Carboniferous itself means coal-bearing. When you burn a lump of coal, you are releasing sunlight that was captured by a lycopsid tree over 300 million years ago. That's not a metaphor. That is literally what's happening. The photons that hit those ancient leaves were converted into chemical energy, locked into carbon compounds, buried, preserved, and you're setting them free. There's something almost dizzying about that connection. But the forests did something else too. Something that matters enormously for our story. All that carbon being pulled from the atmosphere and buried underground meant that atmospheric carbon dioxide levels dropped dramatically during the Carboniferous. And as CO2 dropped, oxygen levels rose. They rose a lot. Today, our atmosphere is about 21% oxygen. During the mid to late Carboniferous, oxygen levels may have reached 35%, maybe even a touch higher. And that changed everything about what was possible for life, especially for arthropods. See, Insects and their relatives don't breathe the way you do. They don't have lungs. They have a system of tiny tubes called tracheae that branch through their bodies, delivering oxygen directly to their tissues by diffusion. It's elegant, but it has a limitation. Diffusion only works efficiently over short distances. In today's atmosphere, that puts a cap on how big an insect can get. But pump the oxygen concentration up to 35%, and suddenly diffusion works over longer distances, and that cap lifts. And the carboniferous arthropods took full advantage. This is the era of Meganeura, a dragonfly-like insect with a wingspan of around 70 centimeters. That's over 2 feet across. Imagine a dragonfly the size of a hawk. There were millipede relatives like Arthroplura that could reach over 2 meters long, the length of a small car. Giant scorpion-like creatures. The carboniferous forests were crawling and buzzing with arthropods on a scale that has never been repeated. Now, into this world of giant insects and towering spore trees, our tetrapod ancestors were diversifying rapidly. And here's where one of the most important splits in vertebrate history happens: the divergence between the amphibian lineage and what would eventually become the amniotes. The reptiles, the mammals, the birds, us. Early carboniferous tetrapods were still tied to the water, the same way modern frogs and salamanders are. They had to lay their eggs in water, or at least in very moist environments, because their eggs didn't have shells. They were soft, permeable, vulnerable. The larvae, think tadpoles, were aquatic. This meant that no matter how well adapted an early tetrapod's legs and lungs became, it could never fully leave the water side. It was tethered. And in the swampy world of the Carboniferous, that wasn't necessarily a problem. Water was everywhere. But it was a limitation, and limitations in biology are invitations for evolution. The amniotic egg is one of those innovations that doesn't sound dramatic but absolutely was. Sometime in the early Carboniferous, maybe around 340 to 320 million years ago, the exact timing is uncertain, and the fossil record of this transition is frustratingly sparse. A lineage of tetrapods evolved an egg with a series of membranes inside it. An amnion, which enclosed the embryo in its own little pond of fluid. A chorion for gas exchange, an allantois for waste storage, and critically, a shell, not necessarily hard like a chicken egg at first, probably leathery like a modern gecko egg, but enough of a barrier to prevent the egg from drying out on land. The amniotic egg was, in effect, a self-contained life support system, a private aquarium for the embryo. It cut the cord to open water, and that changed the trajectory of vertebrate life on this planet. One of the earliest known amniotes is a little creature called Hylonomus, found in the fossil stumps of lepidodendron trees in Joggins, Nova Scotia. These fossils were first described in the 1850s by William Dawson, and they're remarkable. Tiny lizard-like animals, maybe 20 centimeters long, apparently living inside hollow tree stumps, probably feeding on insects and millipedes. The Joggins fossil cliffs are now a UNESCO World Heritage Site, and for good reason. These humble little animals, sheltering inside rotting club moss trunks, were the pioneers of a lineage that would eventually produce dinosaurs, eagles, whales, and you. And here's something worth sitting with. The amniotic egg is, in a real sense, an engineering solution to an environmental constraint. The problem was desiccation. How do you reproduce away from water when your embryo needs a wet environment to develop? The solution was to bring the water with you, sealed inside a membrane. It's the same basic logic that would, hundreds of millions of years later, lead to spacesuits and submarines and pressurized aircraft cabins. you take your environment with you. The amniotes didn't conquer the land by adapting to dryness. They conquered it by refusing to give up wetness, by packaging it, miniaturizing it, making it portable. Now, while the amniotes were making this quiet revolution, the Carboniferous was also the time when the tetrapod body plan was being refined in all sorts of ways. 5 fingers. You've got 5 fingers on each hand, 5 toes on each foot, and so does almost every tetrapod alive today. Frogs, lizards, cats, bats, whales. Whales have 5 fingers hidden inside their flippers. Why 5? Well, the earliest tetrapods didn't always have 5. Remember Acanthostega with its 8 digits, Ichthyostega with 7? In the early Carboniferous, digit counts varied, but fairly quickly, 5 became the standard, and once it locked in, it stuck. There may be something about 5 that's developmentally stable, something about the genetic patterning, the Hox genes that control limb development, that makes 5 a kind of attractor. But honestly, it might also be partly chance. 5 worked well enough, it became the template, and evolution is conservative with templates that work. Every piano concerto ever played, every surgery ever performed, every rock ever climbed, all done with hands built on a pattern that was established in Carboniferous swamps. The Carboniferous is also when the amniotes themselves split into 2 great lineages, and this split matters to you personally because you're a product of one of them. The 2 groups are distinguished by the structure of their skulls, specifically by the number and position of openings behind the eye socket. Temporal fenestrae, they're called. The synapsids had 1 opening on each side. The reptiles, or more precisely the lineage leading to diapsids, had 2. It sounds like a trivial anatomical detail, and in a way it is. These openings were probably related to jaw muscle attachment and flexibility. But this simple structural difference marks the divergence of 2 dynasties. The diapsids would eventually give rise to lizards, snakes, crocodilians, dinosaurs, and birds. The synapsids, and here's the part that surprises a lot of people. The synapsids are our lineage. Mammals are synapsids. You are a synapsid. And in the late Carboniferous and into the Permian period that followed, the synapsids were the dominant large land animals, not the reptiles. The synapsids got there first. The most famous early synapsids are the pelycosaurs, and you've probably seen pictures of the most iconic one, even if you didn't know its name, Dimetrodon. That's the one with the giant sail on its back, the fin-like structure supported by elongated vertebral spines. People often mistake Dimetrodon for a dinosaur, but it's not even close. It lived in the early Permian, around 295 to 270 million years ago, tens of millions of years before the first dinosaurs appeared. And it's more closely related to you than to any dinosaur. Dimetrodon is on your side of the family tree. That sail, by the way, its function is still debated. Thermoregulation is the classic hypothesis, the idea being that Dimetrodon could turn its sail broadside to the morning sun and warm up faster than its prey, giving it a metabolic advantage. Some researchers have questioned whether the sail's structure was really optimized for heat exchange, suggesting it might have been more about display, sexual selection, species recognition. Maybe it was both. Biology rarely does just one thing with a structure. But what's really fascinating about the synapsids is the long, slow transformation from something that looked like a sprawling, scaly, cold-blooded reptile into something warm-blooded, furry, and unmistakably mammalian. And that transformation didn't happen overnight. It played out over more than 100 million years, across the Permian and the Triassic, and it involved changes to nearly every system in the body: the jaw, the teeth, the ear bones, the legs, the metabolism, the skin. It's one of the best documented major transitions in the entire fossil record, and the story of how we know about it involves some truly remarkable fossils and some equally remarkable paleontologists. The jaw alone, the way the synapsid jaw simplified over time, losing bones one by one until only the dentary remained, is a story that connects eating, hearing, and the very essence of what it means to be a mammal. Because here's what happened. In the early synapsids, the pelycosaurs like Dimetrodon, the lower jaw was made up of several bones. Just like in most other vertebrates at the time, you had the dentary at the front, which carried the teeth, but behind it sat a whole collection of smaller bones: the articular, the cerangular, the angular, the splenial, others too, and the jaw joint itself. The hinge where the lower jaw met the skull was formed by two of these bones: the articular on the lower jaw and the quadrate on the upper. This is the ancestral condition. Fish have it. Amphibians have it. Reptiles have it. It's the standard vertebrate jaw joint, and it had been working perfectly well for hundreds of millions of years. But in the synapsid lineage, your lineage, something strange started happening. The dentary bone began to grow. Slowly, generation after generation, million after million years, the dentary expanded backward, taking up more and more of the jaw, while those other bones behind it got smaller, and smaller, and smaller. You can literally line up synapsid fossils from the Permian through the Triassic and watch this happen. It's one of the most beautiful gradual transitions in all of paleontology. The dentary just keeps growing, keeps taking over, until eventually it's doing almost all the work, bearing the teeth, anchoring the muscles, forming most of the jaw's structure. And those other bones? They're shrinking into these tiny little nubbins clinging to the back of the jaw, seemingly useless. Now, you might be thinking, okay, that's interesting in a nerdy anatomical way, but why does it matter? And this is where it gets genuinely wild. Because those tiny bones that were shrinking away from the jaw, they didn't just disappear, they migrated. They moved up into the ear. The articular became the malleus. The quadrate became the incus. And together with the stapes, which was already present as a sound-conducting bone, they formed the three-bone chain of the mammalian middle ear: the hammer, the anvil, and the stirrup. The tiniest bones in your body, right now, sitting inside your skull, amplifying sound vibrations from your eardrum to your inner ear. Those bones used to be part of a jaw joint. They used to help an animal like Dimetrodon bite down on its prey. And now they help you hear music, hear voices, hear the wind. That is exaptation at its most breathtaking. A structure evolved for one purpose being co-opted for something completely different. And we don't have to take this on faith. There are transitional fossils that show the intermediate stages, fossils where these bones are simultaneously part of the jaw joint and beginning to function in hearing. Morganucodon, from the late Triassic and early Jurassic, around 200 million years ago, had what's called a dual jaw joint. The old articular quadrate joint was still there, still functional, but right next to it, the dentary had grown back far enough to also contact the squamosal bone of the skull, forming the new mammalian-style jaw joint, both joints working side by side. The animal could bite with either hinge, and those little articular and quadrate bones were already loosening their connection to the jaw, already beginning their journey toward the ear. It's like catching evolution in the act, frozen in stone. The person who did so much to illuminate this transition was a South African paleontologist named Robert Broom. Working in the early 20th century, Broom spent decades in the Karoo Basin of South Africa. This vast semi-arid expanse of sedimentary rock that turned out to be one of the richest deposits of Permian and Triassic synapsid fossils anywhere on Earth. The Karoo is extraordinary. Layer after layer of ancient floodplain sediments, and embedded in them, thousands upon thousands of synapsid skulls and skeletons. Broom was a character. Trained as a physician, he practiced medicine to fund his real passion, which was fossil hunting. He was prolific, sometimes controversially so, naming new species at a pace that made other paleontologists uneasy. But his work on the therapsids, the more advanced synapsids that succeeded the pelycosaurs, was foundational. He showed the world that the transition from reptile-like synapsids to mammals wasn't a sudden leap, but a long, graded continuum. And the therapsids are where this story really picks up speed. After the pelycosaurs had their run in the early Permian, a new wave of synapsids rose to dominance. The therapsids, appearing around 275 million years ago. These were more sophisticated animals in almost every way. Their legs were starting to move underneath their bodies rather than sprawling out to the sides, which meant more efficient locomotion, less energy wasted. Their teeth were becoming differentiated, not just rows of identical pegs, but distinct incisors, canines, and postcanines, each shaped for a different function. This is called heterodonty, and it's one of the hallmarks of mammals. You have it right now. Run your tongue along your teeth. The flat ones in front for cutting, the pointed ones on the sides for tearing, the broad ones in back for grinding. That specialization started here, in the Permian therapsids. Among the therapsids, one group in particular stands out, the cynodonts. These are the ones that would eventually give rise to true mammals. By the late Permian, cynodonts were already showing a remarkable suite of mammalian features. Some had secondary palates, bony shelves separating the mouth from the nasal passages, which meant they could breathe while eating. Think about why that matters. If you're a cold-blooded animal, you can afford to stop breathing for a while during a meal. Your metabolic rate is low. Your oxygen demand is modest. But if you're evolving toward warm-bloodedness, toward a high metabolic rate that demands constant oxygen, you need to breathe continuously. A secondary palate is an engineering solution to that problem. It's a structural redesign of the skull to support a more energy-intensive way of living. And that's the thread that ties so many of these changes together. The march toward endothermy, toward being warm-blooded. The differentiated teeth allow more efficient food processing, which means more calories extracted per meal. The more upright posture allows more sustained activity. The secondary palate allows continuous breathing. The jaw changes allow stronger, more precise biting. Each modification feeds into the others. It's not that some designer sat down and planned a warm-blooded mammal. It's that each incremental change offered a slight advantage, and those advantages compounded over millions of years, pulling the lineage in a consistent direction. But right in the middle of this extraordinary transformation, something catastrophic happened. The worst mass extinction in Earth's history, the end Permian extinction, about 252 million years ago. And when I say worst, I mean it in the starkest possible terms. Roughly 90 to 96% of all marine species went extinct, around 70% of terrestrial vertebrate species. The planet came closer to being sterilized of complex life than at any other point since the Cambrian. This event is sometimes called the Great Dying, and the name is earned. The cause and this took decades of detective work to piece together, was volcanism on a scale that's almost impossible to comprehend. The Siberian Traps, a massive volcanic province in what is now northern Russia, erupted over a period that may have lasted 1,000,000 years or more, pouring out enough lava to cover an area larger than Western Europe. But it wasn't just the lava. The magma intruded through thick deposits of coal, limestone, and evaporates in the Siberian crust, and those interactions released staggering quantities of carbon dioxide, methane, and sulfur dioxide into the atmosphere. The carbon dioxide drove intense greenhouse warming. Global temperatures may have risen by 10 degrees Celsius or more. The oceans warmed, became acidified, lost oxygen. On land, ecosystems collapsed as temperatures soared and rainfall patterns destabilized. There's evidence of widespread wildfires, soil erosion, fungal blooms feeding on dead vegetation. The world became, for a time, genuinely hostile to complex life. The therapsids were hit hard. Many lineages vanished entirely. The Gorgonopsians, large, saber-toothed predators that had been apex hunters of the late Permian, gone. The Dicynodonts, those strange beaked herbivores that had been among the most abundant land vertebrates on Earth, were devastated, though a few lineages limped through into the Triassic. The cynodonts survived, but barely. The survivors were generally small, possibly burrowing animals, the kind of creatures that could ride out environmental extremes by hiding underground, by needing less food, by being inconspicuous. And here's where the story takes a turn that shapes everything that comes after. Because the Triassic world that emerged from the ashes of the Great Dying was an empty world, ecologically speaking. All those vacated niches, all those open opportunities, and two groups in particular were poised to fill them: the surviving synapsids, the cynodonts and a few dicynodont holdovers, and a new group that was just beginning to diversify: the archosaurs, the lineage that would produce crocodilians, pterosaurs, and dinosaurs. For a while, in the early Triassic, the cynodonts actually did quite well. Some grew large again. Cynognathus, from around 240 million years ago, was a wolf-sized predator with powerful jaws and a body that was looking increasingly mammalian. But the archosaurs were diversifying rapidly too, and by the middle and late Triassic, they were gaining ground. The reasons are complex and still debated. Some researchers point to the archosaurs' respiratory system. Many had air sacs connected to their lungs, similar to modern birds, allowing more efficient oxygen extraction, which might have been crucial in the low oxygen atmosphere of the early Triassic. Others point to posture and locomotion advantages. Whatever the combination of factors, by the late Triassic, the dinosaurs had arrived, and they were taking over. The cynodonts responded by getting smaller. And smaller. This might sound like losing, like retreating, but think about it from another angle. Getting small opened up ecological niches that the big dinosaurs couldn't access. Small crevices, underground burrows, the leaf litter of the forest floor, the canopy of trees. And being small put intense selective pressure on exactly the traits that would define mammals: higher metabolic rates to maintain body temperature at small body sizes, fur for insulation, more efficient food processing, better hearing to detect predators in the dark, nocturnal activity to avoid the daytime dominant dinosaurs. There's a hypothesis called the nocturnal bottleneck, and it suggests that early mammals were pushed into nighttime activity by dinosaur competition, and that this is why mammals as a group have such excellent hearing, such sensitive whiskers, such a well-developed sense of smell, because those are the senses that matter most when you can't rely on vision. When the sun goes down and the world becomes a landscape of shadows and sounds, eyes built for color and detail aren't much help. But ears that can pick up the faintest rustle in the undergrowth, a nose that can read the chemical signature of a predator from dozens of meters away, whiskers that can map the geometry of a burrow entrance in total darkness, those are survival tools. And the fossil evidence actually supports this idea in a really elegant way. If you look at the skulls of early mammals from the Jurassic, the eye sockets tend to be relatively small compared to many of the dinosaurs they lived alongside. But the brain case, the part of the skull that houses the brain, is proportionally larger, and the regions associated with olfaction, with smell processing, are expanded. CT scans of fossils like Morganucodon and another early mammal called Hadrocodium, which lived around 195 million years ago in what's now China, show that the olfactory bulbs were already taking up a significant portion of the brain. Tim Rowe and his colleagues published these CT scan studies in the early 2000s, and what they found was striking. The mammalian brain didn't just get bigger all at once. It expanded in stages, and the first stage was dominated by improvements in smell and touch, exactly the senses you'd prioritize if you were a tiny creature navigating a dangerous world in the dark. And here's what's really lovely about this. You, right now, lying there in the dark, are benefiting from that ancient pressure. The fact that you can hear my voice so clearly, that you can distinguish subtle tones and pitches, that your ears can detect sounds across an enormous range of frequencies. That's a legacy of those Mesozoic nights. Your ancestors didn't choose to become nocturnal. They were pushed into it. But the adaptations they developed in response became some of the defining features of what it means to be a mammal. Now, let's talk about the Mesozoic itself, because for a long time, the standard story about mammals during the age of dinosaurs was pretty dismissive. The narrative went something like this. Mammals spent 150 million years as tiny, shrew-like creatures, cowering in the shadows, waiting for the dinosaurs to go extinct so they could finally have their moment. And that story isn't entirely wrong. Mammals were generally small during the Mesozoic, and the largest dinosaurs were incomprehensibly enormous by comparison. But the idea that mammals were just marking time, just surviving without innovating, has been completely overturned by fossil discoveries in the last few decades, especially from sites in China. The Yanliao biota and the Jehol biota, both from northeastern China, have produced some of the most spectacular mammal fossils ever found. These are Jurassic and early Cretaceous deposits, roughly 160 to 120 million years old, and the preservation is extraordinary. Sometimes you can see the outlines of fur, the contents of stomachs, even the shapes of soft tissue organs. And what these fossils reveal is that Mesozoic mammals were doing far more than just hiding. They were experimenting. They were diversifying. They were filling ecological roles that we never imagined. Take Castorocauda, described in 2006 by G and colleagues. This animal lived about 164 million years ago in the Middle Jurassic, and it was a swimmer. It had a broad, flat tail like a beaver, webbed feet, and dense fur adapted for an aquatic lifestyle. A swimming mammal in the Jurassic, while dinosaurs ruled the land. Or consider Velaticotherium, a mammal from roughly the same period that had a membrane stretched between its limbs. A gliding mammal, like a flying squirrel, soaring between Jurassic trees. There was Repenomamus, from the early Cretaceous, about 130 million years ago, which was the size of a badger and was found with baby dinosaur bones in its stomach. A mammal eating dinosaurs. not exactly the cowering shrew of the old narrative. And then there's Fruitifosaur, from the late Jurassic of Colorado, around 150 million years ago. This little animal had powerful forelimbs and teeth that looked remarkably like those of modern armadillos, suggesting it was a dedicated digger, probably feeding on insects in termite-like colonies. The point is that mammals during the Mesozoic were occupying a surprisingly wide range of ecological niches, swimming, gliding, climbing, digging, even occasionally eating small dinosaurs. They were small, yes, mostly between the size of a shrew and a house cat, but within that size range, they were remarkably diverse. And something else was happening during this long stretch of time, something that would matter enormously later. The major groups of modern mammals were beginning to diverge. Now, this gets into some fairly technical territory, but it's worth understanding the basic layout because it shapes everything that comes after. There are three main groups of living mammals: the monotremes, the marsupials, and the placentals. Monotremes, that's the platypus and the echidnas, are the most ancient branch. They lay eggs, which is the ancestral condition, but they still nurse their young with milk, which is a defining mammalian trait. The split between monotremes and the lineage leading to marsupials and placentals probably happened sometime in the Triassic or early Jurassic, maybe around 200 million years ago, though the molecular and fossil evidence don't perfectly agree on the timing. Marsupials and placentals diverged from each other sometime in the Jurassic or early Cretaceous, roughly 160 to 180 million years ago. The key difference between them, and this is a simplification, but it captures the essence, is reproductive strategy. Marsupials give birth to relatively undeveloped young that continue developing outside the womb, typically in a pouch. Think of a kangaroo joey, born tiny and pink and essentially embryonic, crawling into its mother's pouch to finish growing. Placentals, on the other hand, retain the developing embryo inside the uterus for much longer, nourished by that remarkable organ, the placenta, which allows extended intimate exchange of nutrients and oxygen between mother and offspring. The baby emerges more fully developed. Neither strategy is inherently better. Both have been wildly successful. But they represent fundamentally different solutions to the same problem of how to give your offspring the best start in life. Now, here's something that might surprise you. For most of the Cretaceous, the period from about 145 to 66 million years ago, marsupials were actually more diverse and arguably more successful than placentals in many parts of the world, particularly in North America. The placentals were there too, but they were less prominent. It was the catastrophe at the end of the Cretaceous that reshuffled the deck entirely. But we'll get to that. During the Cretaceous, something else extraordinary was happening in the world around these mammals, something that would reshape terrestrial ecosystems in ways that are still with us today. Flowering plants, angiosperms, were diversifying explosively. The oldest confirmed angiosperm fossils date to the early Cretaceous, around 130 to 140 million years ago. But by the mid-Cretaceous, flowering plants were spreading across every continent, transforming landscapes that had been dominated by conifers, ferns, and cycads. And this wasn't just a botanical event, it was an ecological revolution. Flowering plants co-evolved with insect pollinators, creating intricate networks of mutual dependence: bees and flowers, beetles and blossoms, butterflies and nectar. These relationships generated enormous biodiversity. More kinds of plants meant more kinds of insects, which meant more food for small mammals, which meant more opportunities for mammalian diversification. Think about fruit. Before angiosperms, there was no fruit, no berries, no nuts with fleshy coverings, no sweet pulp designed to entice an animal into eating it and dispersing the seeds. The evolution of fruit created an entirely new food source, and mammals were among the animals that exploited it most enthusiastically. Some researchers think the diversification of primates, our own order, is intimately linked to the spread of fruit-bearing trees in tropical forests. Hands good for grasping branches were also good for handling fruit. Forward-facing eyes, useful for judging distances while leaping between branches, were also useful for spotting ripe fruit among green leaves. But that connection comes later in the story. What matters right now is that by the late Cretaceous, say 70 to 66 million years ago, you had a world that was lush, warm, and teeming with life. Dinosaurs still dominated the large animal niches on every continent. Pterosaurs still ruled many aerial niches. Marine reptiles, mosasaurs, plesiosaurs, patrolled the oceans. But mammals were there, diversifying quietly, refining their physiology, their brains, their reproductive strategies, their senses. They were ready for an opportunity, even though none of them could have known what was coming. And what was coming was the most famous catastrophe in the history of life on Earth. 66 million years ago, give or take, an asteroid roughly 10 to 12 kilometers across, about the size of a small city, struck what is now the Yucatan Peninsula of Mexico, creating the Chicxulub crater. The energy released was on the order of 10 billion Hiroshima bombs, and the immediate effects were almost incomprehensible. Within the first few hours, there was a blast wave, a thermal pulse that may have ignited wildfires across much of North America, and enormous tsunamis radiating outward through the Gulf of Mexico. But the longer-term effects were what really did the killing. The impact launched billions of tons of rock, dust, and sulfur-rich debris into the upper atmosphere, and this material spread around the entire globe. It blocked sunlight. Photosynthesis collapsed. Temperatures plummeted. The food chain, which ultimately depends on plants capturing solar energy, was severed at its base. The Deccan Traps, massive volcanic eruptions in what's now India, were also active around this time, and there's ongoing debate about how much those eruptions contributed to the extinction versus how much was purely the asteroid. Gerda Keller has argued forcefully for a significant volcanic role, while other researchers like Sean Gulick, who helped drill into the Chicxulub crater itself, emphasize the asteroid as the primary trigger. The truth may involve both, but the impact event is the one that lines up most precisely with the extinction boundary in the fossil record. What died? About 75% of all species on Earth, all non-avian dinosaurs, all pterosaurs, all marine reptiles, most of the larger land animals. The ammonites, those beautiful coiled shell creatures that had thrived in the oceans for hundreds of millions of years, gone. It was the end of an era in the most literal sense, the boundary between the Cretaceous and the Paleogene, the Old World and the New. But not everything died, and who survived and why is one of the most important questions in the entire history of life, because the survivors shaped the world you live in now. Small body size helped. Burrowing helped. Being able to eat a wide variety of foods, seeds, insects, detritus, helped, because those food sources could persist even when the photosynthetic base of the food chain was temporarily destroyed. And mammals, by and large, fit that profile. They were small. Many were burrowers. Many were generalist feeders. The same traits that had been shaped by 150 million years of living in the shadow of dinosaurs turned out to be exactly the traits needed to survive the worst day in the history of life on Earth. Think about that irony for a moment. The very things that had kept mammals marginal, their smallness, their hiding, their scrappy opportunism, became the keys to the kingdom. It's one of evolution's deepest lessons, and it plays out over and over again. What looks like a disadvantage in one context can become the ultimate advantage when the context changes. So the asteroid hits, the world burns, then freezes, then slowly clears. And in that clearing, in that strange, emptied-out landscape of the earliest Paleogene, something remarkable begins. The ecological stage has been swept bare. All those niches that dinosaurs had filled for 160 million years. The large herbivore niche, the apex predator niche, the mid-sized browser niche, the aerial niche. They're all suddenly vacant. And mammals, those little survivors, those generalist burrowers and insect eaters, begin to diversify at a pace that's almost dizzying by evolutionary standards. We call it the Paleogene mammalian radiation, and the fossil record shows it happening with startling speed. Within just a few 100,000 years of the extinction boundary, which is practically overnight in geological terms, you start seeing mammals getting bigger. Within a few million years, you see the first large-bodied mammals. Within 10 million years, you've got mammals filling almost every major ecological role on land. It's like watching a garden explode after a wildfire has cleared away all the old growth. The soil is rich, the sunlight is unobstructed, and everything that can grow, grows. Now, there's been a really interesting debate about how this radiation played out. One view, sometimes called the explosive model, says that most of the major mammalian lineages, the groups that would eventually become primates, rodents, carnivores, ungulates, bats, whales, diverged rapidly after the extinction, that the extinction itself was the trigger for all that diversification. The other view, informed a lot by molecular clock studies, suggests that many of these lineages had already split from each other during the Cretaceous, while dinosaurs were still around. But they were all small, all ecologically constrained, all waiting in the wings, so to speak. The extinction didn't create the lineages, it released them. The truth, as best we can tell from combining fossil and molecular evidence, seems to be somewhere in between. Some deep splits, like the one between placentals and marsupials, which we talked about earlier, definitely happened back in the Cretaceous. But the really dramatic ecological diversification, the part where mammals actually started becoming large and filling those empty niches, that does seem to have been triggered by the extinction event. It's the difference between having the genetic potential for diversity and actually expressing it ecologically. The Cretaceous mammals had the potential. The Paleogene gave them the opportunity. And here's where geography starts to matter enormously. Remember, the continents have been drifting this whole time. By the late Cretaceous, Pangaea had long since broken apart. South America and Africa were separating. India was an island drifting northward toward Asia. Australia was pulling away from Antarctica, and these separations meant that different groups of mammals were isolated on different land masses, evolving independently, filling similar niches in completely different ways. It's one of the most beautiful demonstrations of convergent evolution you could ask for. Take South America. After it separated from Africa, and before it reconnected with North America, which wouldn't happen until about 3 million years ago, when the Isthmus of Panama formed, South America was essentially a giant island continent. And on that island, a group of placental mammals called the South American native ungulates evolved into forms that looked astonishingly like horses, camels, and elephants, but were completely unrelated to the actual horses, camels, and elephants evolving on other continents. There was Macrauchenia, this bizarre long-necked creature that looked vaguely like a camel but had a short trunk. There was Toxodon, a massive, hippo-like herbivore that Darwin himself found fossils of and described as perhaps the strangest animal ever discovered. These animals evolved to fill the same ecological roles as their counterparts elsewhere, but from completely different ancestral stock. Same problems, same solutions, different starting points. And then there were the marsupials. In South America, marsupials diversified into predatory forms. The Boreanids and the famous Thylacosmolus, a saber-toothed marsupial predator that evolved its enormous canines completely independently from the saber-toothed cats that wouldn't show up for millions of years. Same ecological pressure, same evolutionary answer. Meanwhile, in Australia, which had become fully isolated even earlier, marsupials underwent their own spectacular radiation, eventually producing everything from kangaroos to koalas to wombats to the thylacine, the Tasmanian tiger, which looked so much like a placental wolf that you'd swear they were related if you didn't know the anatomy. This is convergent evolution at its most jaw-dropping, and it tells you something profound about how natural selection works. It's not random. It's constrained by physics, by biomechanics, by the fundamental challenges of being a certain size in a certain environment eating certain things. There are only so many good solutions to the problem of being a large herbivore on a grassy plain or a fast predator chasing prey through open country. Evolution finds those solutions again and again, independently, on separate continents, with completely different raw materials. It's like different architects in different countries who've never spoken to each other, all arriving at similar building designs because the laws of physics and the needs of the occupants are the same everywhere. But let's come back to our own lineage, because this is where the story gets personal. Among the mammals that survived the extinction and began diversifying in the early Paleogene were the earliest primates. Now, defining exactly what counts as the first primate is tricky. The boundary between early primates and their close relatives is fuzzy in the fossil record, as boundaries usually are. But by about 55 to 56 million years ago, in the warm, forested world of the early Eocene, you've got clear primates showing up in the fossil record. Animals like Tailhardina, a tiny creature about the size of your thumb, found in deposits in Belgium, Wyoming, and China almost simultaneously, which tells you these early primates spread quickly across the northern hemisphere when the continents were still connected by land bridges. And what were these early primates like? Small, arboreal, living in trees, with grasping hands and feet, nails instead of claws, and forward-facing eyes that gave them stereoscopic vision. Every one of those traits is an adaptation to life in the trees. The grasping hands let you grip branches. The nails instead of claws let you manipulate small objects, insects, fruits, seeds, and the forward-facing eyes, giving you overlapping visual fields and depth perception, let you judge distances accurately as you leap from branch to branch. That's your inheritance, by the way. The reason you can thread a needle or catch a ball is because your ancestors needed to grab a branch 40 feet above the ground without misjudging the distance. Depth perception wasn't a luxury. It was survival. And here's something lovely about this. Remember the nocturnal bottleneck we talked about? Those early mammals that developed enhanced smell and touch because they were living in the dark? Well, primates represent a partial reversal of that trend. As they moved into the trees and became more diurnal, more active during the day, vision became paramount again. The primate brain began investing heavily in visual processing, expanding the visual cortex at the expense of the olfactory regions that had been so important to earlier mammals. You can actually trace this shift in the fossil record through endocasts, casts of the inside of the skull that show brain shape. Matt Cartmill proposed what's called the visual predation hypothesis back in the 1970s, suggesting that the forward-facing eyes of primates evolved specifically for catching insects on thin branches in a visually complex forest environment. It's a compelling idea, and while the details are debated, the broad picture holds. Primate evolution is, in many ways, the story of becoming visual creatures again after millions of years in the dark. The Eocene was a golden age for primates. The climate was warm, much warmer than today. There were palm trees in Alaska, crocodiles in the Arctic. Dense tropical and subtropical forests stretched across much of the globe, and primates thrived in those forests. Two major groups emerged: the adipoids, which are related to modern lemurs and lorises, and the omomyids, which are closer to the lineage that would eventually produce monkeys, apes, and us. These early primates were still small, still mostly insect eaters or fruit eaters, but they were diversifying, experimenting with different body plans and ecological strategies. And then the climate began to change. Starting around 34 million years ago, at the boundary between the Eocene and the Oligocene, the Earth entered a significant cooling phase. Antarctica, which had been connected to South America by a narrow land bridge, finally separated completely, allowing the Antarctic circumpolar current to form. That current essentially isolated Antarctica thermally, letting it cool dramatically. Ice sheets began forming on Antarctica for the first time in tens of millions of years. Global temperatures dropped. Forests retreated. Grasslands began to expand. This cooling had enormous consequences for primates. In the northern hemisphere, in North America and Europe, where primates had been abundant during the warm Eocene, they largely disappeared. The forests they depended on shrank and fragmented, and primates, being tropical and subtropical animals, couldn't cope. The future of primate evolution would play out primarily in Africa, Asia, and South America. And it's in Africa, during the Oligocene, around 30 to 35 million years ago, that we find some of the most important fossils in our own ancestry. The Fayum Depression in Egypt, today a dry, dusty landscape southwest of Cairo, was, in the Oligocene, a lush tropical forest on the edge of an ancient sea. And in those Fayum deposits, paleontologists like Elwyn Simons spent decades excavating fossils of early higher primates, the anthropoids. Creatures like Egyptopithecus, sometimes called the dawn ape, a small, monkey-like animal about the size of a house cat, with a relatively large brain for its body size, and the beginnings of the dental pattern that would characterize later apes and humans. Egyptopithecus lived about 29 to 30 million years ago, and while it's not a direct ancestor of ours, the precise lineages are still being worked out. It gives us a window into what the common ancestors of Old World monkeys and apes might have looked like. From animals like these, two great lineages diverged: the Cercopithecoids, which became the Old World monkeys, baboons, macaques, colobus monkeys, and the hominoids, which became the apes. And the apes, during the early and middle Miocene, roughly 23 to 10 million years ago, had their own spectacular radiation. This is something most people don't realize. When you think of apes today, you think of a handful of species: gorillas, chimpanzees, bonobos, orangutans, gibbons, and us. A small, rather beleaguered group, mostly endangered. But during the Miocene, apes were everywhere. They were the dominant primates across Africa, Europe, and Asia. Dozens of species, filling ecological roles that monkeys fill today. Proconsul in East Africa, a tailless, fruit-eating ape from about 18 million years ago. Dryopithecus in Europe, Civipithecus in Asia, which appears to be closely related to the ancestors of orangutans. The Miocene was the age of apes. But then the climate shifted again. Continued cooling and drying through the late Miocene shrank the forests further. Grasslands and woodlands expanded across Africa and Eurasia, and the apes, which were mostly forest-adapted, fruit-eating animals, began to decline. Monkeys, which were generally better at eating leaves and other lower-quality foods, began to replace them ecologically. The apes retreated to shrinking forest refugia. Most lineages went extinct. But in Africa, in those drying, fragmenting forests and expanding woodlands, one lineage of apes began to do something different. Instead of retreating deeper into the remaining forest, they started adapting to life at the forest edge, in the mosaic habitats where woodland met grassland, where trees were scattered rather than continuous. And somewhere around 6 to 7 million years ago, this lineage split. One branch would eventually lead to chimpanzees and bonobos. The other branch, our branch, would step out onto a path that led, through millions of years of experimentation and adaptation and extinction and survival, toward something unprecedented in the history of life on this planet. And the first thing that changed, the very first thing that set our lineage apart from our closest relatives, wasn't our brains. It wasn't our tools. It wasn't our language. It was the way we walked, the way we moved across the ground. Bipedalism. Standing upright on two legs and walking, one foot in front of the other, with our hands free and our spine vertical, and our eyes scanning the horizon instead of the forest canopy. That was the founding adaptation of our lineage. And it happened long, long before we became anything you'd recognize as human. Now, here's what's fascinating about that. If you asked most people what makes humans special, they'd probably say our big brains, or our ability to talk, to think abstractly, to build cities and write symphonies and argue on the internet. And all of that's true. All of that matters. But those things came much, much later. The fossil record is very clear on this. For millions of years, our ancestors walked upright, with brains no bigger than a chimpanzee's. Bipedalism came first. The brain expansion came second. And the gap between those two events is enormous. We're talking 3 to 4 million years of upright walking before brain size really started to take off. So whatever story you want to tell about human evolution, it has to start with walking. It has to start with the feet. And the earliest evidence we have for this comes from some truly remarkable fossils. One of the oldest known hominins, and I should mention, the term hominin refers to us and all our relatives after the split from the chimpanzee lineage. One of the oldest is Sahelanthropus chadensis, found in Chad in 2001 by a team led by Michel Brunet. The fossil is a crushed but recognizable skull, nicknamed Tumai, and it dates to roughly 6 to 7 million years ago, right at the boundary of that split we were just talking about. The skull has a curious mix of features: a small, ape-sized brain, heavy brow ridges, but a foramen magnum. That's the hole at the base of the skull where the spinal cord exits, positioned more underneath the skull than behind it. That positioning matters because it suggests the head was balanced on top of a vertical spine, rather than jutting forward from a horizontal one. It hints strongly at upright posture. Though there's debate. There's always debate with fossils this old and this fragmentary. Some researchers have questioned whether Sahelanthropus was really a hominin at all, or perhaps a relative of the gorilla lineage. but the position of that foramen magnum is suggestive. Then there's Ardipithecus, specifically Ardipithecus ramidus, known from fossils in Ethiopia dating to about 4.5 million years ago. The most famous specimen is called Ardi, and she was described in detail in 2009 by Tim White and his team after 15 years of painstaking preparation and analysis. 15 years. The fossils were so fragile, so crushed by millions of years of sediment, that extracting and reconstructing them was an extraordinary feat of patience. And what Ardi showed was something nobody quite expected. She was a biped, yes. Her pelvis was broad and short, in a way that supported upright walking, very different from the long, narrow pelvis of a chimpanzee. But her feet still had a grasping big toe, divergent from the other toes, clearly useful for climbing trees. She lived in a wooded environment, not on open grasslands. So Ardi was a creature that walked upright on the ground, but could still climb effectively in the trees. A mosaic, a transitional form in the truest sense, living in exactly the kind of mixed habitat we'd expect. That forest edge, those scattered woodlands where our lineage was experimenting with a new way of moving. And this tells you something important about bipedalism. It didn't evolve on the open savanna. That was the old story, the one you might have learned in school. That our ancestors came down from the trees because the forests disappeared, found themselves on the grasslands, and stood up to see over the tall grass. It's a neat story, but it doesn't match the evidence. The earliest bipeds lived in wooded environments. They stood up among the trees, not after leaving them. The reasons for bipedalism are still debated, and honestly, it was probably multiple factors working together. Freeing the hands to carry food, especially to carry it back to a partner or offspring. More efficient long-distance locomotion. And this is a real thing. Bipedal walking is actually more energy efficient than quadrupedal knuckle walking over long distances, which matters when food sources are spread out. Thermoregulation. Standing upright in the tropical sun exposes less of your body surface to direct overhead sunlight and raises more of your body into the breeze. There are arguments about display, about weighting, about all sorts of things. The truth is probably that several of these advantages reinforced each other. Evolution doesn't need one reason. It just needs a slight edge, compounded over generations. By about 3 to 4 million years ago, bipedalism was fully established. And we know this not just from bones, but from one of the most hauntingly beautiful pieces of evidence in all of paleontology, the Laetoli footprints. Discovered in Tanzania in 1978 by Mary Leakey's team, these are actual footprints preserved in volcanic ash that had been dampened by rain and then hardened. 2, possibly 3 individuals walking side by side across the landscape about 3.6 million years ago. The prints show a clear arch, a non-divergent big toe, a heel strike followed by a toe off, essentially a modern human gait. No knuckle impressions, no sign of a grasping foot, just 2 upright walkers strolling across a volcanic plain in what is now East Africa, 3.5 million years before anyone would chip a stone tool. The species most commonly associated with this time period is Australopithecus afarensis, and the most famous individual is Lucy, discovered in 1974 in Ethiopia by Donald Johansen and Tom Gray. Lucy is about 3.2 million years old, stood maybe 3.5 feet tall, and had a brain roughly the size of a chimpanzee's, about 400 cubic centimeters. But from the waist down, she was remarkably human-like. Her pelvis, her knee joint, the angle of her femur, all adapted for efficient bipedal walking. From the waist up, she was more ape-like, with long arms relative to her legs and curved fingers that suggest she still spent significant time in the trees. Sleeping in trees, perhaps, escaping predators. But she walked. She walked upright across the African landscape, and she did it well. And here's where the story starts to accelerate, just a little. Because for a long time, from Sahelanthropus at 6 or 7 million years ago through the Australopithecines at 2 to 3 million years ago, brain size didn't change much. These were small-brained bipeds living in Africa, eating mostly plant foods, probably some insects, maybe scavenging meat occasionally. They were successful. Multiple species of Australopithecines coexisted across Africa. Some, like Australopithecus africanus in South Africa, were more gracile. Others, like Paranthropus boisei in East Africa, developed massive jaws and enormous chewing muscles for processing tough, hard foods, roots, tubers, seeds. Paranthropus had a sagittal crest on top of its skull, a ridge of bone for anchoring those powerful jaw muscles. It's sometimes called Nutcracker Man, a specialist, and specialists are vulnerable when conditions change. But around 2.5 to 2 million years ago, something shifted. We start to see the first stone tools in the archaeological record. The Oldowan industry, simple cobbles with a few flakes knocked off to create a sharp edge. For a long time, these were attributed to early Homo, but recent discoveries suggest Australopithecines may have been making simple tools too. The oldest known stone tools, from Lomekwi in Kenya, date to about 3.3 million years ago, which is before any known Homo species. So tool use, like bipedalism, may have preceded the genus Homo. But it was within our genus that tool use really took off. The emergence of Homo is messy. The fossil record between 2 and 3 million years ago is complicated, with multiple species that are hard to sort out. Homo habilis, Homo rudolfensis, possibly others. What's clear is that around 2 million years ago, brain size started to increase. Homo habilis had a brain of about 600 to 700 cubic centimeters, noticeably larger than the Australopithecines. And then came Homo erectus, appearing around 1.9 million years ago. And this is where things really change. Homo erectus had a brain of roughly 900 cubic centimeters or more, was tall, long-legged, with body proportions much more like modern humans. Homo erectus was the first hominin to leave Africa, spreading into Asia and possibly southern Europe. Fossils from Dmanisi in Georgia date to about 1.8 million years ago. Java man, Peking man. These are Homo erectus. This species persisted for well over 1 million years, making it arguably the most successful hominin species ever in terms of longevity. And Homo erectus was associated with a new, more sophisticated stone tool technology, the Acheulean hand axe. Beautifully symmetrical, teardrop-shaped tools that required planning and skill to produce. Some of them are genuinely lovely objects. And Homo erectus controlled fire, though exactly when fire was first deliberately used is debated. Evidence from Wonderwork Cave in South Africa suggests controlled fire around 1 million years ago. Fire changed everything. Cooking food externally meant less energy spent on digestion, which meant more energy available for the brain. It extended the day. It provided warmth, protection from predators, a social focal point. Think about what a campfire does to a group of people even today. It draws them together. It creates a space for storytelling, for bonding, for sharing. And the brain kept growing. Through the Middle Pleistocene, various populations of Homo across Africa, Europe, and Asia continued to evolve. In Europe, one lineage adapted to the cold, glacial conditions, and became the Neanderthals, Homo neanderthalensis, with brains actually slightly larger than ours on average, around 14 to 1500 cubic centimeters, stocky bodies conserving heat, sophisticated stone tools, burial of their dead, possible symbolic behavior. In Asia, another lineage became the Denisovans, known mostly from DNA extracted from fragments of bone found in a Siberian cave, a discovery that would have seemed like science fiction just 30 years ago. And in Africa, our own species, Homo sapiens, emerged sometime around 300,000 years ago, based on fossils from Jebel Irhoud in Morocco described by Jean-Jacques Hublin and colleagues in 2017. And here's what's wild about this. For most of our existence as a species, we weren't alone. We shared the planet with Neanderthals, Denisovans, possibly late-surviving Homo erectus in Southeast Asia, Homo floresiensis, the so-called hobbits of Flores Island, tiny hominins with small brains who made tools and hunted small elephants, surviving until perhaps 50,000 years ago. Homo naledi in South Africa, with its puzzling mix of primitive and modern features. The human family tree isn't a tree at all. It's a braided stream with populations splitting and merging and exchanging genes. We know from ancient DNA that Homo sapiens interbred with Neanderthals. If you have ancestry from outside sub-Saharan Africa, you carry roughly 1 to 2% Neanderthal DNA right now, tonight, in the cells of your body as you lie here. Some of those Neanderthal genes affect your immune system, your skin, your hair. They're functional. They were useful. They were kept by natural selection because they helped our ancestors survive in new environments. And then, somewhere between about 70 and 50,000 years ago, something happened. Homo sapiens began to spread out of Africa in a major way, reaching Australia by at least 65,000 years ago, which required crossing open ocean. Think about that. Think about the courage or desperation or curiosity that drove people onto rafts or boats with no certainty of land ahead. Reaching Europe by about 45,000 years ago, where they encountered and eventually replaced the Neanderthals. Reaching the Americas by at least 15,000 years ago, possibly earlier. And everywhere they went, they brought with them symbolic thought, art, language, complex social structures, the cave paintings at Lascaux and Chauvet, the carved figurines, the shell beads found at Blombos Cave in South Africa, dating to 75,000 years ago, music, ritual, story. And that brings us, after this long, long journey, to something worth sitting with for a moment. You, here, now, listening to this. Every single thing we've talked about tonight, the formation of the Earth, the first self-replicating molecules in some warm volcanic pool, the billions of years of bacterial life, the great oxygen catastrophe, the first eukaryotic cells swallowing their future mitochondria, the invention of sex, the snowball Earth and the Cambrian explosion, the fish that crawled onto land, the synapsids that survived the Great Dying, the tiny mammals hiding in the dark while dinosaurs ruled, the asteroid that ended one world and began another, the primates leaping through Eocene forests, the apes that stood up and walked. All of that is in you. It's not metaphor, it's literal. Your cells still run on the same ATP chemistry that the earliest life used. Your mitochondria still carry their own DNA, a remnant of that ancient symbiosis. The bones in your middle ear were once jaw bones in a Triassic therapsid. Your spine is a modified fish spine. Your hands are modified fins. You breathe air because a fish developed a swim bladder that became a lung. You see in color because a primate needed to find ripe fruit in a green canopy. You are the current edition of a manuscript that has been continuously edited for nearly 4 billion years. Not one generation was skipped. Not one link in that chain was broken. Every one of your ancestors, stretching back to the very first cell, survived long enough to reproduce. Every single one. Through ice ages and asteroid impacts and supervolcanic eruptions and predators and diseases and famines. The odds against you being here, being exactly you, are so staggeringly large that they're essentially meaningless as a number. And yet, here you are. So as you lie there, and your breathing slows, and the details of everything we've talked about tonight start to soften and blur at the edges the way things do when sleep is close, just hold on to that one thought if you can. You are not separate from the story of life on Earth. You are the story. You're the latest sentence in a narrative that started with chemistry and starlight and water, and every chapter is still written in your bones, your blood, your DNA. The whole history of life is keeping you warm right now, keeping your heart beating, turning the air in your lungs into energy the same way it's been done since the very beginning. And that's enough. That's more than enough. Let it all go now. Let the names and the dates and the millions of years dissolve into something quieter. Something that just feels like the deep, old, patient rhythm of life doing what it has always done. Continuing. Changing. Finding a way. You're part of that rhythm tonight. You always have been. Rest now. The story will still be there in the morning.